for joining Portrait Pharmaceuticals as we discuss the various aspects of method validation. Today, we will be focusing on linearity. First, we will go over why linearity is performed and how it is practically performed in the laboratory. Then we will discuss what the ICH requirements are and how the various types of applications or instrumentation affect your validation range and acceptance criteria. Finally, we will discuss a few things that might need to be considered when developing your linearity experiment for your validation. Linearity is the direct proportion of the concentration of your analyte to the response of the detector. This simply means that if the concentration of your sample doubles, then the response obtained by the detector doubles as well. It is important to show this correlation for most methods used in pharmaceutical analysis, as the concentration or content is measured by some type of detection source. A response is most commonly the peak area of the analyte in a chromatogram obtained by HPLC or GC. Other responses can be absorbance values, volumes of titrant, and others. Most methods need to show a linear response. Linearity is typically performed across a range of nominal concentration of the analyte and should be wider than the specification. For instance, the specification for an assay method is 90 to 110% label claim. The linearity would be performed from 80 to 120% of the label claim, ensuring that the method is still linear outside of the typical sample concentrations. The ICH guidelines require 5 levels over the tested range. For quantitative analysis, a correlation coefficient must be not less than 0.99. A common mistake we see labs and analysts make is to report the coefficient of determination, which is R squared. The ICH guidelines specifies R. Additionally, linearity must be demonstrated over the range in which impurities will be reported, usually from the limit of quantitation to 120% of the specification or 2% of nominal. This linearity is performed and prepared in the same manner as the upper level linearity. When preparing linearity solutions, a single stock should be prepared and all dilutions performed from that single stock. Serial dilutions may also be used. If material is available and quantities of solvents or solutions are not restricted, a good rule of thumb is for your stock to be 10 times the concentration of your nominal or 100% level. This makes the dilutions easy to manage and plan for with typical glassware sizes. If using this route, ensure that the analyte is soluble at that concentration prior to getting started. In the case that I have shown here, quantities are limited, but solubility is not. The linearity stock solution is prepared at 1 mg per ml, and 20 mL is prepared. We like to confirm through an Excel calculation that our proposed concentrations are correct. So our second column is our target concentration, and the last column is the actual concentration using the dilution scheme specified in the third and fourth columns. Also make sure your dilution scheme does not use more than your available stock volume. The total volume in column 3 is 17 mils. We prepared a 20 mil stock, so we have enough to perform all dilutions from that same stock. It is highly recommended to inject each linearity solution in duplicate. Now that we know how to perform, here is a table showing when linearity is required. This is a table that was derived from the ICH guidelines indicating when and what methods require linearity. As you can see, the phase of your project, as well as the type of assay that you are doing, affects this decision. For a limits test, for a phase 1 program, linearity is not required, but for a phase 2 and beyond, linearity must be proven. For all phases, the assay method should be linear and therefore will be required for those methods. Some additional considerations to take into account when planning your linearity experiments. You must know the solubility of your compound in the diluent or solvent that you are using. You may use a neat solvent or other diluent that the compound is more soluble in for your stock solution preparation, and then prepare your dilutions in the final diluent. As you enter into the later phase development, the quantitation of impurities and degradants are critical. So you need to make sure that you are reporting them correctly. Some impurities have a different response than the active at the same detection wavelength. This difference in response must be accounted for in the calculation. Commonly, low-level linearity of the impurity is performed. The slope of the curve is then divided by the slope of the upper-level linearity curve for the active. If this ratio is outside of the range of 0.8 to 1.2, then this ratio should be included in the calculation of the impurity result. Finally, some active ingredients are salts. For example, this is lidocaine HCl. In solution, the HCl dissociates from the lidocaine molecule, and the detector is only providing response for the lidocaine. The label claim of your drug product could be represented as lidocaine or lidocaine HCl. If the label claim is for lidocaine, then a molecular weight correction must be performed so that your range of your linearity curve accurately reflects the range of the label claim to be tested. This concludes our overview of linearity. If you have any questions, please feel free to reach out. Portrait Pharmaceuticals is an analytical testing laboratory focusing on small molecules. We have a unique process flow that allows us to provide results within half the time of similar laboratories. Looking forward to hearing from you. Thanks so much.